Aging cultures are maintained in test tubes at 30 degrees Celsius with constant agitation by a roller drum. An aliquot of cells from each aging culture is inoculated into a well of a bioscreen honeycomb plate containing YEPD medium. The bioscreen CMBR machine incubates and shakes the honeycomb plate for 24 hours while measuring optical density of each well every 30 minutes. Growth curves can be generated from the data output by the bioscreen, which can be used to calculate viability of each aging culture. After the 24-hour incubation, cell densities in the honeycomb plate wells can vary depending on the viability of the original aging culture.
Hi, I'm Chris Murakami from the laboratory of Dr. Matt Kaeberlein in the Department of Pathology here at the University of Washington. Today we're going to show you a procedure for quantifying yeast chronological lifespan using outgrowth of aged cells. We use this procedure in our lab to study how both genetic and environmental factors influence the longevity of yeast. So let's get started.
To prepare aging cultures, first, streak the yeast strains from frozen stocks onto YEPD agar plates. Incubate the cells at 30 degrees Celsius for 48 hours or until single colonies appear.